Good afternoon, everybody, and welcome to this new episode of EFSA's webinar on protein safety assessments in GMOs. It is a great pleasure to welcome you here and listen to how we adapt our risk assessment to food innovation and what needs to be done in terms of research and development for the future of filling methodological gaps. In previous webinars already, we described how we started this work from experiences in our GM plant dossiers and how we funded 7 important research projects to gather further knowledge. And gradually, we augmented the collaboration with our applicants, our member states partners, and we also reached out to international risk assessment communities. Today, we are glad to present the results of the knowledge as published over this summer and the results of this work. My name is Renilde Schoonjans. I have the pleasure to be team leader of GMO Food and Feed Safety Assessment, and I'm the moderator of our webinar today. So thank you all very much for joining and showing such great interest in our work. We have received over 300 registrations representing food industry, universities, international organizations, NGOs, and national competent authorities from Europe and beyond. From EFSA's side, there is a team here of colleagues. First, our 2 expert presenters on the topic, which are Antonio Fernandez, Scientific Officer in the Nutrition and Food Innovation Unit, and Javier Moreno, who is the chair of the GMO Food and Feed Working Group. As a Q&A manager for today, Telemachos Gumperis will be helping me and Martina and Ariana with handling your questions that you can submit throughout the webinar in the Q&A feature. And now, a bit on the webinar structure. Antonio will introduce you in his presentation of about 15 minutes to the reasons and the objectives of our activity on protein safety assessment. Javier will continue by explaining in more detail the Outlook section, which contains next steps for filling the knowledge gaps, and that might inspire research groups after this webinar to take up some of the suggestions. And then follows our live Q&A session for about 20 minutes using the questions posted by you. As some housekeeping rules are necessary, we want to notify that you are automatically connected in a listening mode only, and that we would like to ask you to submit questions in English only, and that this event will be recorded. It will be published together with the presentations on the EFSA webinar shortly after this event. You can submit your questions in the Q&A feature, which you find on the right-hand side of the presentation window, and you can do this throughout the entire event. And Telemachos will aggregate them and pass them to the respective speakers to be addressed during the Q&A session. So we'd like to ask you to keep the questions short, because we have limited time, and in scope of scientific risk assessment. That means that clarifications about authorization process or any other legal aspects are outside the scope of this webinar, and we will not address them here. But we also want to tell you that if some questions remain unanswered due to limited time, you can resubmit them via the Ask EFSA service that you can find on the efsa.connect platform. And with this, I would like to give the floor to our first presenter, Antonio Fernandez, Scientific Officer in the NIF unit.
Thank you, Renilde, and good morning, good afternoon to everyone, and thank you for joining to this event. This is with great pleasure to start the presentation of this work of the EFSA GMO panel, that we don't consider it as the end, but the starting of a productive journey to continue. So I will initiate with some background and then move into the terms of reference of the document together with Javier. Whenever we will talk about protein safety, the scope of this document has been protein toxicity and protein allergenicity. In terms of risk assessment, Codex define the principles that has been embedded in all documentations around the globe, including Europe, that took into account all the elements considered for the risk assessment, including knowledge on the protein source, bioinformatic analysis, in vitro studies, and in vivo studies. However, we should all take into account that all these elements were set with the standards in 2003. Taking this into account, the scientific community has urged the modernization of protein safety assessments. What EFSA has achieved with this scientific opinion is a reflection on current practice, challenges, and future opportunities on protein safety in GMOs, and has done that following a terms of reference defined in the document, 4 terms of reference. Terms of reference 1, lessons learned from experience in the assessment of newly expressed proteins in the last 20 years. Terms of reference 2, building upon the experience, new methodologies available with the potential to be used as complementary and or alternative to the current testing strategies. Terms of reference 3, roadmap for future implementation of complementary alternative methods. And terms of reference 4, recommendations for further development, could also be implementation, and research activities. So the GMO panel has took into consideration 3 main aspects. One, the experience gain. The other one, developmental projects. And finally, engagement. I will tackle one by one briefly. First, starting with the experience gained, the GMO panel has produced around 200 scientific opinions on GM plants and has already assessed more than 40 newly expressed proteins. This is showing that the safety assessment of GMO plant has been done throughout the years with sufficient robustness, but there are challenges ahead of us. And this has been one of the main aspects has been highlighted in this document. And this relates to the newly expressed protein nature. We are receiving applications with proteins that are difficult to extract and purified, challenging the current approaches, or GM plants with a high number of proteins. The GMO panel has also identified the need to move with criteria for important terms, such as history of safe use, what is meant by similarity, or thresholds, how to apply those to newly expressed proteins. And finally, the need to develop complementary or alternative methods. In this regard, it brings the question on how to validate those or have them with regulatory acceptance. Next, please. Yeah. The second aspect considered by the GMO panel to produce this document has been developmental projects run by external contractors that has been published mainly last year. And these were related to allergenicity assessment, developing novel approaches to increase reliability of predictions, toxicity assessment, exploring in silico and in vitro tools, effects of processing, the relevance for protein safety, development of adverse outcome pathways specific for celiac disease. Also on this topic, a tool was developed for peptide binding prediction, and finally, methodologies for open reading frames. Last, EFSA also took into account the engagement from 3 main means. We had a series of webinars, as described by Renilda before, to ensure that the EFSA scientific work and opinions are clear and open to external scrutiny. We had different stakeholders meetings to foster diverse perspective and promote inclusivity in the work. And finally, we ran a public consultation where the feedback gathered helped EFSA to enhance the quality, accuracy, and relevance of its scientific assessments. This all to make sure that EFSA scientific work is grounded in robust scientific evidence, and for which we thank you all very much for your contributions. So now, starting with the first part of the work, terms of reference 1, lessons learned and complex cases. In the document, we made an effort to include different examples to describe how the current approach has been applied to the different newly expressed proteins. And it's important to highlight here that for the proteins evaluated so far, these are typically show simple enzymatic activity. Most of them are water soluble, and overall, they do not present major challenges for purification and characterization. This makes it not so difficult to follow a current approach as described in the figure. This is what the EFSA GMO panel has been doing, requiring information on the molecular biochemical characterization, bioinformatic analysis, in vitro protein degradation, effect of temperature and pH, and on a case-by-case basis, moving into animal studies when required for toxicology or human sera testing. However, the question remains whether this is the current state of the art in the assessment. Can we do our assessment more proportionate and effective? And the question number 2 comes from complex cases for which the current approach is very difficult, if not impossible, to be carried out. And for this one, we included there some examples like proteins difficult to produce and isolate, membrane-bound proteins. We already have applications on this. Proteins with potential bioinformatic hits with toxins and allergens, for which we also have a few examples. GM plants with numerous proteins to assess. And finally, we don't have examples here for the future proteins that are new to nature. With this, I will stop my presentation. Thank you very much, and I pass the floor to Javier.
Okay, thank you, Antonio. Hi, everyone. Let me now take you to the terms of reference 2. Here we focus on new approach methodologies, especially in silico and in vitro tools. These methods, as you probably know, are becoming more and more available in research, but when it comes to regulatory risk assessment, their use is still limited for several reasons. The highlighted box in yellow in this slide summarizes the principal barriers. Validated targeted in vitro assays for regulatory use are lacking. Existing frameworks remain strongly rooted in a chemical risk paradigm that is not fully fit for multi-protein GM plants. There is no comprehensive guidance for interpreting novel in silico outcomes, and standardized protocols and interpretation criteria for in vitro readouts, for instance, digestion conditions that reflect realistic exposures, are still needed. Turning to the 3 columns below, advanced in silico tools could rank allergenicity, could apply machine learning for toxicity screening, compare 3-dimensional structures and model functional similarity. Also, it's possible to place proteins in phylogenetic context and even predict aggregation. But the utility depends on fit-for-purpose databases and agreed interpretation criteria. In vitro advances include improved mass spectrometry for protein characterization, combined in silico in vitro assays for enzyme specificity, and refined gastrointestinal digestion protocols to identify fragments of potential relevance. These methods should be hypothesis-driven and have standardized readouts. For in vivo testing, the recommendation is tiered and case-by-case. Animal studies should be used only when predicted hazards cannot be resolved otherwise, and any such study must be targeted to fill a specific data gap and designed to minimize animal use. Importantly, exposure could be somehow integrated across all tiers of assessment. Estimated exposure is not yet systematically used for protein safety assessment, but it could critically change the outcome of our risk characterization. Looking ahead, the way forward is to promote discussion and reach consensus on the regulatory role of new approach methodologies by embedding them into an integrated tiered framework, starting with in silico, moving to in vitro, and resorting to in vivo studies only when necessary. We could make the assessment of novel proteins more proportionate, science-based, and future-proof. Next slide, please. The term of reference 3 outlines the key aspects for implementing complementary and alternative methods within risk assessment strategies in the future. The proposed roadmap reinforces the weight of evidence approach in a structured, stepwise manner, prioritizing in silico and in vitro methodologies, and reserving animal studies for cases where hypotheses must be tested or where critical safety data are missing. As you can see in the figure on the slide, this framework is clear and proportionate, is also flexible, and can be applied case by case. It's meant to provide clear decision criteria for protein safety assessment. We start at the top of the figure by asking a simple question, which is the number 1. Is the safety of the newly expressed protein or donor organism described in the literature? If solid evidence exists, we move to the next step. If not, we follow an alternative evaluation path right from the beginning in the right side of the slide that I will explain later on. If we move to the left side of the figure, when evidence is available to the first question, the next checkpoint is the history of safe use, number 2 in this slide. If the protein or its source has a clear record of safe use, the assessment obviously becomes simple. We then compare the sequence identity, which is the number 3, with proteins already known to be safe. Of course, a 100% match strongly supports safety and usually means little additional work needed. But as similarity decreases, the level of scrutiny can increase with, for instance, biochemical or functional assays, structural analysis, or targeted comparison to toxin and allergens, depending on the case. If the history of safe use is not clear, the concept of read across can be applied. Here is the number 4 in the center of the figure. That means using information from well-studied proteins to predict properties of the less well-known proteins which is under study. In practice, if a novel protein is closely related to a safe, well-characterized protein, this read across can sometimes justify waiving some experiments if the supporting evidence is strong. And finally, when no prior safety data exists, the roadmap directs us to targeted in vitro studies to the right side of the figure. This can include stability and digestion tests under gastrointestinal-like conditions, as well as studies on the effects of pH and temperature. If these studies reveal no hazards and give us sufficient information, further testing may not be needed. But if hazard, we are now in the question number 5, if hazards appears or can be predicted or critical data are still missing, the roadmap allows us to escalate with additional in vitro work and reserve animal studies for targeted needs. In the opinion, this approach has been tested on both real and hypothetical case studies. Overall, this approach seems to be practical, ethical, and adaptable across different protein types and functions. Next, please. Now we will turn to term of reference 4, which covers a recommendation for further method development and research to address needs. The GMO panel has proposed, as you can see here in this slide, a set of ranked priorities to focus resources on the activities likely to have the most important impact in our opinion. I will start by covering the 3 core priorities, which range from short to long term. First, as you can see here, history of safe use and biological read across. This is the top short-term priority because having a robust shared approach here could help further reduce testing. What we need is a clear, fit-for-purpose definition of history of safe use and read across for protein safety, including, for instance, measurable criteria like generation of exposure, minimum consumption thresholds, and population characterization. Key actions could be to convene expert working group, agree on relevant criteria, and build an open database of comparator proteins with metadata on exposure and study quality. Second, we have the in silico methodologies in the short, medium term. The current approaches to screening could benefit from modernization, modern in silico approaches. We might need to update sequence and structure similarity thresholds, collect validated fit-for-purpose databases for toxins, allergens, and known safe proteins, and create a standardized framework to interpret in silico outcomes. Also, it's important the exploration of how advanced computational tools, meaning by that 3-dimensional structural prediction, phylogeny approaches, or also artificial intelligence-driven similarity metrics, we need to explore how all these tools can reinforce read across justification. Third, we have the in vitro methods. In the medium long term, we could modernize digestion and other assays so they better reflect human or animal physiology and key toxicological pathways. Priority activities include testing harmonized gastrointestinal-like digestion protocols, defining interpretation criteria for digestibility readouts. In the past, EFSA already identified some potential elements like peptides, minimum peptide cutoffs, persistence and abundance, and also qualifying novel alternative methods for relevant pathways of harm. We think that also proof of concepts are important to show the proof of concept projects are important to show the utility of these in vitro strategies in real risk assessment scenarios. Next slide, please. We are still within the term of reference 4, and beyond the core activities that I mentioned in the previous slide, there are additional priorities that should follow as resources alone, and that may be raised up after further stakeholder engagement. First, we have in vivo testing. Where animal studies remain necessary, they could be hypothesis-driven and targeted, applied on a case-by-case basis. Ideally, all work should pursue the 3 R's model, reduction, refinement, and replacement, and focus on more informative, protein-specific study design. Second, we would like to explore in the future exposure and thresholds. We should expand research on occurrence, dose-response relationships, and exposure estimation. This work could enable the establishment of a scientifically justified threshold for toxicology and allergenicity, and levels. This threshold could be some levels below which no adverse effects are really expected. Third, post-market monitoring could be important to develop hypothesis-driven PMM systems to confirm exposure assumptions or to reduce residual uncertainties from pre-market assessment. Fourth, fundamental research on de novo sensitization. Predicted new onset allergenicity remains a major knowledge gap. Targeted basic research is required to better understand the mechanisms of sensitization and how preclinical signal could be translated to actual clinical risk. Fifth, it's important to study processing and food matrices. I mean, how common food processing steps could affect protein stability, toxicity, allergenicity, and digestibility, because processing can, let's say, change risk. Six, omics approaches. It's important to investigate where and how omics data can add value and develop guidance on interpretation so that such complex data sets can be used robustly in a weight of evidence framework. To put this into practice, we need for sure multidisciplinary expert groups, stakeholder engagement, also funding for proof of concept projects, open and well-curated databases, and agreement on interpretation criteria. Together, these steps will make the protein safety assessments more transparent, reproducible, and balanced. Next slide, please. I think it's the last one. Yes. So to conclude, the primary purpose of this scientific opinion has been to critically evaluate the overarching framework for safety assessment of newly expressed proteins. Our goal has been to highlight the key challenges and gaps that have emerged based on the experience during these last decades of the GMO panel, and to explore practical approaches for addressing them in the near to medium term through targeted implementation and research activities. Looking ahead, the GMO panel's immediate priorities are clear: to reach consensus on operational criteria for history-safe use and read across, and on how to select appropriate comparator proteins to perform this type of studies. Also, it's important to develop fit-for-purpose databases and validation criteria for novel in silico tools, and for example, to support robust protein similarity and homology assessment. These steps could help make assessment more consistent, evidence-based, and proportional. Finally, we want to invite you to contribute to the next phase of work. As you can see in this slide, EFSA will host an online workshop on protein safety prediction and speed in risk assessment on the 12th and 13th of November. The aim is to shape recommendations to improve protein safety prediction and to streamline risk assessment workflows. So please join us, bring your practical case studies or questions or comments, and help build the consensus and tools we need to make these priorities a reality. And that's all. I will now give the floor to Renilde, and thank you very much for your attention.
Thank you very much, Javier and Antonio. You have highlighted very well the key elements of our work from the past. And I think we have reached a milestone with this publication to make very clear what are our priorities in terms of core activities for the future. And so with this Q&A that I now open up, please, audience, see it as an opportunity to ask us further details of what you have been seeing in the presentations, or share already some ideas for that future work. And note also in November, there's another engagement for this. I see that we have meanwhile lined up already a nice number of questions, and there is also a couple of recurrent ones. So I pass the floor to Antonio for the first question. Thank you very much.
Thank you very much, Renilde, and thank you, Javier. Indeed, Renilda, we had a series of recurring questions throughout the process that somehow are linked to the overall goal, the two main goals we try to achieve. First, address the complex cases, and second, have proposals on how to perform our risk assessment in a more proportionate and effective manner. Linked to this, the two questions we selected here, we had as recurring questions was one, could new proposals be used in ongoing applications? Will it be compatible with current requirements? And here there are a couple of keywords we wanted to emphasize today. One is related to flexibility, and flexibility will be considered whenever a strong, high-quality data is provided. And the second keyword we also wanted to use that on a case-by-case basis and within the weight of evidence approach, the strong argumentation together with the strong scientific justification will always be taken into consideration for the overall safety assessment. We in the document included some examples of ongoing applications for which we have employed some of the recommendations already described in the document. But we also wanted to highlight that in some cases, for some NEPs, this might not always be possible, and we will still need to have this dialogue all together on how to perform safety assessment for, as mentioned before, those complex cases. So this will not be the end of the story, as mentioned at the beginning, but it's a path that we are taking to continue. The second question we receive from you is, will there be harmonization with other protein safety legislative frameworks or with other international authorities? And this is somehow also linked to some of the questions we receive here related to the concept on history of safe use. Does the production method play a role? Naturally produced proteins versus protein fermentation? And here an aspect we wanted to highlight is that in this document that we produce, we provided a state-of-the-art focus on newly expressed proteins assessment, providing avenues on how to continue in the future performing these assessments. And this document took as a starting point Codex Alimentarius, defined 25 years ago, but it's also important to say that it goes beyond that document. And we focus on modernization and potential ideas for the future implementation. And here is where several aspects of our document, and here I mean not only the approaches we describe, but also the challenges we saw, could also be relevant for other domains. And this, for example, could be, as mentioned before, the criteria of history of safe use that we don't have yet, and would be advisable to have one that could be used interdomain. The need for fit-for-purpose databases and better integration of bioinformatics tools into the overall safety assessment. With this, I give the floor to Javier, maybe to tackle one of the questions on the number of proteins and to continue with the Q&A. Thank you.
Okay, thank you, Antonio. Yes, I can see there is a direct question about, can you give an example of high number of protein that was considered challenging? Okay, I can tell you that we have a specific example in the opinion. It's an hypothetical GM plan expressing multiple unknown membrane-bound proteins. I would say there is not a fixed number, but if there are a sufficient high number of NEPs, this can compromise from a practical point of view the current risk assessment strategy, which is normally based for GM plants expressing a low number of newly expressed protein, and also to perform individual to assess the proteins in an individual way. So I will say that for hard to isolate proteins or GMOs with many new proteins, we need maybe let's say, smarter or more practical strategies, relying more on in silico and in vitro to try to deal with this type of crops. And the second question that, yes, I can also address is, can you give an example where omics have been useful in risk assessment? As far as I recall, I think we have received some application dealing, for instance, with proteomics to quantify endogenous soybean allergens. And this was based on LC coupled with tandem mass spectrometry, and the applicant provided a very comprehensive picture of the main proteins present in the crop. I don't remember by heart if we have received any other transcriptomic or metabolomics studies. I don't remember, but for instance, I think there is some potential to use some targeted metabolomics. For instance, I think this also could be feasible to be incorporated into the risk assessment. And now I will give the floor back to Antonio.
Thank you, Javier. I will continue with the next couple of questions related to the OSU, read across, and maybe something about post-market monitoring as well. And here is somehow linked to what are also the next steps that were defined in the presentation, throughout the presentation, and maybe summarize in a few words here from me. So in this document, EFSA has framed potential gaps and challenges where improvement is needed. These are those defined by you: history of safe use, post-market monitoring, read across, how to apply that for proteins. And we also highlighted how novel technologies could be used in the weight of evidence to address these gaps, but more work is needed. And more work is needed. We define this prioritization in three main clouds. The first one was related to a history of safe use, read across, and phylogeny. And here we will have further discussions in November, as mentioned before, in a workshop that will be organized by EFSA. And the EFSA GMO panel is also discussing what additional activities will be needed to pursue this aspect. Obviously, the definition of history of safe use is not an easy task. We don't know also how far it will take us. It has been there for many years without a definition, so maybe it's something complex to be performed, but probably we can define criteria that can be used for the safety assessment. The second key aspect was to define how to better integrate novel bioinformatic tools into the safety assessment. And third, the potential use of in vitro testing, targeted in vitro testing. In relation to the comment on post-market monitoring, obviously that's also an aspect that is quite difficult to tackle, as there is yet, as mentioned before, not a protocol to be followed. But whenever there are indications of potential adverse effects or likelihood of allergenicity, regulation asks for post-market monitoring, and applicants are asked to follow those recommendations and suggest post-market monitoring. Whenever provided, this will be assessed accordingly. As mentioned before, just to wrap up, the next two important next steps will be the November EFSA workshop and the EFSA GMO panel following activities. And this will need for sure a stakeholder engagement with you again on how to pursue these activities. I will give the floor now to Javier to continue with the questions. Thank you.
Okay, yes, I will check some question. Will you strive for criteria for read across? It will most likely need to be a case-by-case basis. Will it be possible to have a pre-submission consultation to discuss the need for specific studies? For example, if the GFL, it could be very impactful to the approval timeline if an applicant does not believe a study is needed, that is then requested by EFSA. Okay, so I mean, my reply goes in the same direction, and as Antonio mentioned before, I think this is a critical point, the concept of history of use, read across that in the document we highlight this is needed an international consensus. Currently, as far as we know, an workable operational definition is lacking. And to focus on read across, obviously read across can work when a new protein is clearly related to one we already know is safe. And if the relationship is strong enough, we can sometimes rely on that evidence instead of repeating new experiments. I mean, this is the idea behind to introduce this concept, but again, we need further discussion. It's not an easy task, but I think that there are new advances, especially in situ advances, that can somehow reinforce this potential read across, for instance, to take advantage of phylogeny, or as is included in the opinion, artificial intelligence-based structural clustering, etc. But for sure, this is going to be one of the points that is going to be tackled. Regarding pre-submission consultation, to be honest, I don't have the answer to this technical question. So now I will give the floor back to Antonio if there are other questions to be replied.
Yeah, thank you, Javier. If I may just answer also a little bit more on the pre-submission advice. As you know, EFSA at the moment does not do pre-submission advice, but with this type of documents, as the one we publish here on protein safety, we aim to provide further clarity on how this assessment could be performed in the future whenever complex cases or you want to apply novel technologies into your NAPs. So this is one of the points I wanted to say. There was also a question related to the number of scientific opinion published, over around 240 newly expressed proteins assessed. However, whenever these proteins are further included in a stack or a new crop, extensive risk assessment is still being requested. I think it's important to answer to this question, meaning that whenever we have the same level of information as in an original dossier of an app, EFSA is not requesting for additional information of those proteins. So if this protein has been assessed and you provided the relevant information that made the panel able to conclude on that protein, this information is not requested again to the applicants. How do you see the balance on data requirements on such events versus complete new events? This is what I wanted to highlight here. And for stack applications, EFSA does not reassess those newly expressed proteins, but mainly involve bioinformatic analysis. There was a question on the stakeholders and how they can participate in the workshop in November. If at the moment we have how is it called, the pre-notification of the event, just to pencil this event in your calendar, and very soon we will publish the link to register to this. It will be open to the public, so anyone can register to that. And we are planning to have different breakout sessions to tackle three main aspects. One related to history of safe use, read across phylogeny. Another bulk to tackle in vitro and in vivo testing, and another one to tackle inter-domain relationship. So I hope with this we will further proceed the questions we have for this workshop event, And the idea is to have an EFSA event report that will bring future vision for the future. I will give back the floor to Javier. I don't know if you have more. Yeah, we can give you the floor, Javier.
Okay, thank you, Antonio. Yes, I can see just one. Even if one GM protein is safe, what about the ecological and human microbiome safe from replacing traditional crops with GM monocultures? Why is ecosystem level nutrition excluded from protein safety? Are we pretending the body is isolated from its agricultural environment? This is a great question, and also tricky one that I think goes beyond the GM field. For sure, I think it's important to take into consideration the impact or also the mitigating effect of the gut microbiome. Indeed, EFSA already started to work in that direction by finding several thematic grants dealing with the human and animal gut microbiome, and also with the soil and plant and environment microbiomes. Obviously, there is still some limitation in the knowledge of the microbiome, especially on the functions of the microbiome, but there are huge efforts to trying to bridge the gap between the microbiome research and the risk assessment. Meaning by that, to trying to produce a standardized and adequate method to evaluate the potential impact of not only the proteins, any type of dietary modulator or food additive or contaminant or pesticide metabolites. This is first. It's also important to agree on a minimum requirement or minimum level of requirements to really assess this potential impact. And of course, there are also other collateral problems that complicate this issue, like the individual variability, lifestyle factors, and other factors. So this is a very interesting question, which EFSA is working on it, but I think it goes beyond the impact of the GM plants. So thank you. I don't know if there are any other questions, Antonio, that you would like to address. Okay, I will give the floor to you.
Thank you, Javier. A couple of more questions. There was one related to more focus on allergy. Why is there still no predictive model for whether GM proteins can cause new allergies? Current practice only check for allergen databases. Isn't this like checking the path? Okay, yeah. So related to this, indeed, there is no validated method yet existing, one that can fully predict allergy. I think this is all well known internationally, not only for EFSA, but all around the world. And indeed, all authorities are using a weight of evidence approach for that. As mentioned during the presentation before, EFSA commissioned an external report to an external contractor to develop new ideas for future to improve this predictability of allergy. And there was some promising ideas on how to include clinical relevance in a more prominent way within the whole allergenicity assessment. This is something that will be discussed further, I would say, in the workshop in November and in the future activities of the GMO panel, how these ideas could improve the way we perform allergenicity assessments at the moment. Yeah, there was also another comment related to this on the term safety and how does it impact for populations that are already allergic. And here, maybe we can say, in particular for GMOs, there is also the assessment within the allergenicity assessment of endogenous allergenicity that is done in a comparative manner. So indeed, the assessment is in that aspect for endogenous allergens is not linked to allergenicity in absolute terms, but how does compare to products that are already known to be allergenic. This is specifically done for soybean, and our assessment concludes whether or not the overall allergenicity has been changed. So this information is also considered by the GMO panel. Yeah, there was also another comment, and I will finish here and give you the floor, Javier. It was related to validation. I'm just reading now. It is good to know EFSA commitment to consider validations. This approach will contribute to more reliable and accurate outcomes. I'm curious whether these will be opportunities for industry to participate in this validation process. Indeed, as mentioned before, all these aspects cannot be tackled by EFSA alone, I would say, and for sure stakeholders and international engagement, as we have been doing also maybe in the context of OECD, will help to tackle this aspect of validation, or if you wish, regulatory acceptance that was discussed also at that moment at the OECD context. So thank you very much to all. I will give back the floor to Javier. I don't know if there are more questions there. There are 3-4 minutes left. Thank you to all.
Okay, Antonio, yes, I can take it just a couple of them. There is another one related to exposure. If the exposure role is not clearly defined, how reliable are ADI conclusions? Is it honest to publish safety opinions when the entire dose response framework is still unfinished? Well, I think we don't set ADIs for GM proteins in the way that we can do for chemical. For newly expressed proteins, risk conclusions rely on a weight of evidence approach, meaning by that hazard identification, biochemical functional data, in silico in vitro evidence, and exposure estimates when available, using conservative assumption and clear uncertainty statements to inform whether further testing or mitigation is needed. So in that chain, I think that if there is some uncertainty, the opinion will call for targeted additional data, restricted use condition, or hypothesis-driven post-market monitoring. And there is another more practical question: if will the November workshop offer in-person participation, or will it be virtual only? As far as I know, I think it will be only virtual. So just stay tuned, because for sure EFSA will announce the workshop, and it would be great if all of you could participate. And I think that's all from our side. I think we are approaching to the end of the webinar. So I will give the floor back to Renilde to close the meeting. Thank you very much for your interesting questions. Thank you.
Yes, thank you very much. There was a very lively Q&A, for which I really thank the audience. It shows a great level of interest. And unfortunately, it is indeed time to close this webinar. And let me start by acknowledging all the people here on slide that made it happen. As said, it is a series, so let's see what the future holds. But we do this as a team. It requires some preparation, but I think it's a very useful moment to check in with us and ask us questions. So thanks again for this lively Q&A session. And in general, it was a great pleasure to have you here listening today. We are impressed with the high level of your interest. An important take-home message is that we have, until today, our experiences with the safety testing of newly expressed proteins and the GM plant itself. It shows from over the past 20 years that we have learned about the general safety of these products for consumers, animals, and the environment. But now, in the phase of more innovative products and also new traits, we need practical modernization, as explained so well by the speakers. So we are here to reach out, and we are conveying that new assessment approaches are needed, but we also need all stakeholders to collaborate on this joint future. Should you have important questions or concerns that we were not able to address today due to time constraints, you can still post them on the via Ask a Question on the EFSA Connect platform. And again, the recording of this webinar will be available alongside with the presentation. Also, you can help us by giving your 5 minutes to fill in the evaluation survey that you will receive per email after we close this webinar. But also, last but not least, you may want to consider to subscribe to the newsletter, because that channel will notify you automatically on upcoming stakeholders initiatives and the provisions to enroll. So for instance, the November 1 is a workshop with experts to bring this work further with practical collaborations and with real-life examples from innovative products of genetically modified products or beyond that come to the market. So we are looking forward to welcome also your scientific advancements and your experiences in that forum on protein safety assessment. So with this, I close the webinar and wish you all a very good day. Bye-bye.